Tina Hessman-Say. I'm a senior writer at Science News magazine, and I cover molecular biology.
And I'm Erin Garcia de Jesus, a general assignment reporter at Science News. But since January of 2020, Tina and I have mostly been covering the coronavirus and COVID-19.
We're here because Science News is turning 100, and as part of our centennial celebration, we're talking to scientists about some big ideas in their fields. We have with us now 4 experts, 4 scientists whose research is helping us learn to combat the coronavirus, and I'll let them introduce themselves.
Hi, I'm Mark Dennison. I'm a professor of pediatrics at Vanderbilt University Medical Center in Nashville, Tennessee, and I have been studying coronaviruses, I guess, since 1984, mostly focusing on their replication targets, and more recently on antivirals.
Hi, my name is Akiko Iwasaki. I'm a professor of immunobiology at the Yale University School of Medicine. I'm also an investigator of the Howard Hughes Medical Institute. My lab studies antiviral immunity. Prior to studying SARS-CoV-2, we were studying different viruses, including herpes simplex virus, influenza, Zika, rhinovirus, and others. And since March of 2020, we pivoted to studying SARS-CoV-2 infection and immunity.
Hi, my name is David Martinez. I'm a postdoctoral scholar in the Barrick Lab at the University of North Carolina. In addition to that, I am a Hannah Gray Fellow of the Howard Hughes Medical Institute. And prior to COVID-19, then I had also studied viral immunity to a number of different viruses, including HIV and dengue virus as a flavivirus model. And at the start of the COVID-19 epidemic, then our lab rapidly shifted our focus to this novel coronavirus, and we've been studying the immunity to SARS coronavirus 2 and even SARS-related viruses and just the greater beta coronavirus genus ever since.
Hi, I'm Kanta Subbarao. I'm the director of a WHO Collaborating Centre for Reference and Research on Influenza in Melbourne, Australia, and I'm a professor in the Department of Microbiology and Immunology at the University of Melbourne. I was previously at the NIH for many years. I've worked on influenza for most of my career and started working on SARS actually with the great introduction by Mark Dennison back in 2002 when I was at the NIH, and we had a very active SARS program for about 5 years working with the Barrick Lab and with Mark. And about 5 years ago, I moved to Australia, where I primarily work on influenza, but of course, since the emergence of SARS coronavirus 2, we've been working on SARS 2 as well.
Fantastic. Thanks all of you for those great introductions of yourselves. We're so excited to learn and hear what you guys have to say today. Akiko, our first question is actually for you. So you mentioned that before the pandemic, you were not working on coronaviruses. You pivoted in March 2020. And then I was wondering if you could tell us a little bit about what that was like to pivot your research.
Yeah, so this was quite abrupt, as you can imagine. Until, as I said, March of 2020, we were working on a variety of different viruses and also cancer immunology, and everyone was doing their own thing. And of course, the school was shut down for research other than those that are dedicated to studying SARS-CoV-2. And of course, as a viral immunologist, I was very motivated to study this new coronavirus and trying to understand the immune responses that are happening in the patients. And so we set up a biorepository called Impact Yale, where we would collect longitudinal biospecimen from patients in the hospital. And that's how we sort of started to study immune responses as they happen in the patients. And every day was an excitement because we learned, all kinds of things on a daily basis. And at the same time, it was very scary because we didn't have a vaccine at the time. I wanted to make sure my lab members are safe. We took every precaution we can, and thankfully none of us got infected. but it was also a scary time for everyone.
Well, Mark, you obviously were studying coronaviruses, and you mentioned that you've been working on some antivirals. Maybe you can talk a little bit about how we have developed treatments against the coronavirus and where you see that going next.
Oh, let's see. This is what do we have? Like 12 hours? Is that what we have here? Well, our focus had really been on the viral polymerase and on the novel proofreading exonuclease that coronaviruses encode, which was really first described in a virus infection in my lab back in 2007. And that had sort of led me to interacting with other scientists, particularly polymerase biologists like Olga Pearson at Colorado State and Craig Cameron, who was at Penn State at the time, and saying because this enzyme seemed to be knocking out the capacity of nucleoside analogs to inhibit coronaviruses, we wanted to try to find compounds that would work just really biochemically or genetically, to really inhibit the virus replication. So we had been really working on fundamental principles of replication, and starting then around 2013, we established our collaboration with Gilead and with our long-term collaboration with UNC. And when we discovered that this compound, which we didn't know about what it was at first, was because we were working with it blindly, was remdesivir, and showed that this inhibited the virus so dramatically, we then really pivoted back starting in 2014 to thinking about the capacity of these to actually inhibit coronaviruses broadly. We were interested at the time, of course, in MERS and using our model viruses as well, and SARS, and so SARS-CoV and then MERS, and working at these and showing this. And I think our enthusiasm just really sort of grew and grew as these first remdesivir and then the drug EIDD 1931 that became molnupiravir both showed this really impressive, dramatic capacity to bypass this proofreading function and inhibit the virus replication. I started learning what I call learning drug development as a second language late in life, where I wasn't very smart, but I was really enthusiastic. And we were also seeing the capacity of these viruses, of other bad viruses, et cetera, to infect human cells. So we started sort of pounding the drum and standing up on the street corner and preaching that pandemics were coming and that we needed to be prepared for them. And so we were really, it just positioned us to be able to do that. And publishing that work right before this started, I think, was just I think it's unusual to call it serendipity in the face of a pandemic, but we're grateful that we were in a position to do that. So I think where we're going is, right now, obviously, the NIH and the whole world is really interested in this issue of particularly orally available antivirals targeting multiple different targets that can be broadly active against coronaviruses, at least the family of coronaviruses, if not more broadly against other viruses as well. So I think this renewed emphasis and sort of this dramatic focus around antivirals is important, because I think we're seeing that as good as the vaccines are going to be, we're still going to have people who are at risk, people who are immunocompromised, people who need either prophylaxis or they need treatment. And that's something that we're very excited to continue to try to probe.
Right, and so, I mean, there's been, obviously, remdesivir is now approved for treating COVID-19 for people who are in the hospital, and molnupiravir, which I learned recently is named for Thor's hammer.
Well, that's maybe apocryphal, but that's what I was told by one of the people who developed it. Mjolnir is Thor's hammer, so that's where I heard it came from.
Right. So anyway, that has recently had some encouraging results in clinical trials for people who have been newly diagnosed. Mm-hmm. Why didn't we have these antivirals before? I mean, we've had coronaviruses around.
Well, I don't know that there was more than one or two funded programs in the world working on coronavirus antivirals before the pandemic. We may have been the only one in the United States that I'm aware of that was focused specifically on that. I think part of it is because antivirals are fundamentally difficult. They're more difficult than vaccines, so apologies to vaccinologists. And the reason for that is because viruses, a vaccine can work outside of a cell. An antiviral has to work inside of a cell, since viruses are obligate intracellular parasites, and they use host cell mechanisms. So anything you do with an antiviral fundamentally is going to be targeting the inside of a cell and mechanisms that may be common to the cell and the virus. So, and also viruses are exceptionally good at evading these, and finding something that can damage the virus without damaging the cell is profoundly challenging. I think maybe a final answer is because there had never been sort of a Manhattan Project type approach where everybody in the world was looking at all their compounds, was really willing to target this, and that the resources were brought to bear to bring immunologists, animal model geneticists, virologists, biochemists, structural biologists, evolutionary biologists together to try to understand quickly, do we have targets that could work, and how do we develop those rapidly?
Right. Well, I want to switch over and talk to Kanta a little bit about, well, this is a pandemic, it's global, and you're in Australia on the other side of the world from the rest of us who are in the United States. Um, can you tell us a little bit about what the experience has been like in Australia and for the rest of the world? Because you've been one of the people who have been monitoring the spread of the virus, and what are you seeing, especially in terms of variants and what we can expect out of this coronavirus.
Sure. I think that the experience of the pandemic has been very different in different parts of the world. Australia is a huge island, and they closed their borders very early. We closed our borders in February of last year completely. So there was very little introduction of the virus into Australia. And the same was true in New Zealand, in Taiwan, and Hong Kong, Singapore. A lot of countries closed their borders. So their experience of the pandemic is really quite different from what the US, the UK, Europe, and so on saw. What we also did in Australia was they instituted a mandatory 2-week quarantine for anybody returning to the country or coming into the country. So that 2-week period was actually long enough to essentially capture any infection that was brought in. So seeding of the community just wasn't happening very much. It did happen when we had breakdowns in the hotel quarantine system, which did happen. But we've had, we were really protected through the first year and something of this pandemic. However, the Delta variant has really changed all of that because it is far more infectious. And I don't know if it's partly pandemic fatigue. Australians have been incredibly compliant, I must say, having spent most of my adult life in America. Wearing a mask was just not a political statement. It was just done because we were asked to do it. And people were very compliant with all of the restrictions. So the country did very well. Unfortunately, now, of course, we're dealing with the fact that, Delta is here and it's spreading. We're dealing with the fact that Australia got a little complacent and instituted and actually deployed vaccine rather slowly. So we were pretty late to the party for vaccines, and only now are really catching up. So now we've overtaken the United States in the number that are fully vaccinated. And again, maybe it's the fact that Aussies are a little more compliant, but in many parts of the country, more than 80%, well over 80% of the population has had their first dose and will get their second dose within the next few weeks. So I think we will reach, and Canberra, which is the country's capital, has over 99% first vaccination. So they will be the most vaccinated city in the world probably soon. So, it's been quite different, and it's been a little heartbreaking for me to watch the pandemic transpire in the US. I worked for the US government for most of my adult life, and it's been really quite devastating to watch the size of the pandemic and the fact that, there was such a breakdown in the support for public health over the years that, essentially this pandemic was such a full-blown event in the United States. And even now when the numbers are coming under control, it's still far greater than, Australia's ever seen through the whole pandemic. So in a sense, sitting far away, it's been a bit of a mixed bag of how I feel about it. I mean, a lot of people here say, "Oh, aren't you just so glad you're here?" And I think, no, everybody I know is in America, and it's very hard to be far away. My family's there, so it's hard to be far away and observe this from a distance. But on the other hand, in terms of science, it's been amazing, um, and for so much, I spend a huge number of my nighttime hours on Zoom calls and phone calls, and the progress of science has been, just breathtaking.
Yeah, Kanta, you mentioned the Delta variant. I'm curious what your thoughts are about, things were looking better before the Delta variant came. There were other variants as well that people were concerned about. Like, what are your thoughts on how things would have gone had that, like, particularly the Delta variant not come on the
So
scene?
that's a good question. I mean, it's a hypothetical because, of course, the Delta variant is very much here and has taken over all over the world. And I don't think it's going to be the last variant we see. I think we will see additional variants emerge and I think the reason we're seeing variants is because of the burden of infection in the population around the world. And we could talk about vaccine equity and third doses and so on, but until we vaccinate more of the world, we're going to see infections in unvaccinated people and the emergence of variants. So without the Delta variant, the vaccines were looking fantastic. And they continue to do very well in protection against severe disease. So if the initial goal of vaccination was to prevent severe illness and death and to protect our healthcare systems. And they've done very well with that, even with the Delta variant. Now, I think if the Beta variant had actually taken off and spread around the world, we would have been in even worse trouble because the Beta variant actually is less well inhibited by the antibody elicited by the original Wuhan strain. Against the Delta variant, I think the big issue is that it is highly infectious and that even people that are fully vaccinated can get infected. Now, I think it is not entirely clear yet how much a fully vaccinated person who gets infected with the Delta variant, what is the risk of them actually spreading it to other fully vaccinated people? I think that is a piece of data we don't have yet. We do know that vaccine-induced immunity, the antibody titers are dropping with time. We know that the Delta variant is very fit and is spreading. We also know that fully vaccinated people can get infected with the Delta variant, and they can shed virus, at least by PCR, at a similar load as an unvaccinated person. But the bit of data that I don't see yet is how many fully vaccinated people a fully vaccinated person can infect. So I think our hope is to actually get the level of vaccination up in the world so that we can drive down some of this burden of infection.
Speaking of vaccines and variants, this brings me to David. You and your colleagues are working on, like next generation vaccines, thinking about the future of a world where we have lots of different variants or even other emerging coronaviruses, and working on something that could tackle all of those in a universal coronavirus vaccine. What do you think have we learned about the potential for these types of shots from your work?
Yeah, so I think so far, based on all the data we've seen, then achieving at least a more broad vaccine, at least against the cousins of SARS coronavirus 2, might be a more readily achievable thing to do. So we've been able to at least preclinically develop vaccine concepts against SARS-related viruses, including SARS coronavirus 2 and the critical variants, such as the beta variant that Kanta mentioned, that is relatively more neutralization resistant compared to other variants. So in terms of the broad spectrum vaccines against the SARS-related viruses or the sarbecoviruses, then I think that might be a easier target to achieve. So we have a couple of different vaccine concepts that we've been involved with, and so far, both of those concepts appear to be highly protective against a number of different of those viral infections. I think moving forward, what will be a more difficult target to hit will be a more broad or a beta coronavirus vaccine, for example, that not only covers the SARS related viruses, but also includes, for example, group 2C coronaviruses, which include MERS, or even the common cold coronaviruses.
So you mean we might not have to have colds anymore?
That would be ideal, and that of course remains an aspirational goal. But I think moving forward, that is certainly where we would all like to get in terms of developing a beta coronavirus widely protective vaccine that could not only uh, prevent the infection from SARS coronavirus 2, but also more distant uh, coronaviruses. But of course, that is currently being tested preclinically. So we don't yet know if that'll be possible in humans.
Can you talk a little bit about what makes these universal vaccines different from the ones that we have now against the coronavirus? I mean, most of them are directed against the spike protein, or they are killed whole viruses. Um, is that kind of approach you're taking? Like you get a, a spike protein that is one size fits all, or are you doing a different type of approach?
So a number of different approaches are being tried. We've been involved in at least two of them, and one of the approaches that we've been primarily involved in is actually expanding the breadth of the spike by admixing the different immunogenic domains, so the NTD, the RBD, and the S2, in generating chimeric spikes. And then with the advent of the mRNA technology, then you're able to multiplex the delivery of these vaccines. So that is one approach that has shown immense early success, at least in mice so far. And in another approach that we've also been involved in is the delivery of a nanoparticle vaccine that expresses the RBD of SARS-related viruses. And the RBD of SARS coronavirus 2, for example, actually shares a highly conserved signature with a number of different SARS-related viruses. So we've actually identified monoclonal antibodies that are broadly protective against a number of different SARS-related viruses. And if you deliver the part of the RBD that has this broadly protective epitope, then you can readily induce broadly protective immunity in mice. So what's important and what's promising is that you have at least you have more than one platforms that can achieve this. And I think that raises optimism for me.
Yeah. Well, Akiko, we've been talking about vaccine immunity, but you've been studying a lot about what goes wrong in the body, or what just happens in the body when you get infected with the coronavirus. Can you talk a little bit about what you've been learning, and how is this virus response different than other coronaviruses or just other respiratory viruses?
Yeah, so one of the first learnings that we got from studying these patients was that there is this, in the severe cases, the patients are generally unable to control the virus well, especially quick enough. And what happens is that once the patients fail to do so, the virus spreads through into the lung and damage some of the very delicate tissues that are required for oxygen, gas exchange, and so on. And that's sort of leading to this pneumonia phenotype. And when we study those patients, what we're finding is that there is misfiring of the immune response, that normally we think of antiviral immunity mediated by one branch of the immune response, which is called the type 1 immunity. But what we're seeing is that those patients are also engaging type 2 and type 3 immunity, which are normally reserved for fighting parasite infection or fungal infection or bacterial infection. And so, what appears to us is sort of like a misfiring of the immune response, that trying to contain this virus, with any tools that we have on hand, but actually that's resulting in a pathological outcome in some of these patients because it's not a well-intended, antiviral mechanism. So that was the first insight we obtained from studying the patients in real time. And later on, we also studied sex differences in immune responses that occur in these patients. And, as we all know, male sex is a risk factor for severe and lethal COVID. And what we learned is that in the early phase of the immune response, women tended to generate more robust T cell immunity, whereas men failed to do so, but they instead engaged inflammatory responses. And that may have led to sort of the lack of control of the virus that leads to this misfiring later on that I just mentioned. So we also learned that there's a difference in the way male and female immune responses are dealing with the virus, and that may explain to some extent some of the, different risk factors that we're seeing based on sex.
I mean, there's also a lot of people have been talking more about long-term effects of viral infections and long-term symptoms that you might have after you've recovered from the virus infecting your body. It's supposedly gone, but you still have a lot of symptoms. Long COVID, and I know you've been really involved in that particular field as well. So can you talk a little bit about what you've learned in that arena?
Yeah, absolutely. So that's what I'm most concerned about right now is the lack of diagnosis and care and therapy that can help people with long COVID. And so long COVID can happen in a large fraction of hospitalized patients who are discharged alive. Anywhere estimate between 50 to 70% of these people are still having lingering symptoms after months of discharge. And long COVID can also happen in people who had milder infection who were never hospitalized. But those patients may also develop long COVID. The estimated percentages are varies, but between 5 to 30%. So even if we take a conservative estimate of 5%, that's a large number of people given how many millions of people are infected with this virus. And the latter type is also, there are more women in middle age that's enriched in that population. And so we currently have a couple of hypotheses we're trying to dissect. One is that the long COVID can happen as a result of potentially a viral reservoir or some fragments of virus RNA or protein that deposits in some tissue that's causing chronic inflammation. The other hypothesis we're pursuing is that this acute infection triggers autoimmune disease, which can have long-term consequences, which involve multiple organ systems. And so long COVID has at least 200 different symptoms that have been reported involving like pretty much every organ system you can imagine. And so we really need to find out what's going on in these patients so we can treat them better.
Are there good treatments at this point?
Now, I wish there were some good treatments. I'm not sure if the antivirals would actually work in these patients. If it is actually a persistent virus infection, that might have some, beneficial effect. But we have to remember the long COVID is likely going to be a heterogeneous disease, that it's not one thing that's causing all these symptoms. So if we can identify, let's say, a cluster of patients that may be driven by persistent virus versus those that are driven by autoimmunity or a combination thereof, we can start to think about rational therapy for these people. But first is really to identify what's going on so we can have a better therapy. And the one thing that seems to be helping patients is the vaccination, actually. So there are now reports demonstrating that, between 40 to 60% of the long haulers that receive the vaccine feel improvement in their symptoms, whereas about 20% are getting worse symptoms. So that gives us a little bit of idea as to what kind of triggers might improve their symptoms and, what we can do to help them.
So I have a question. Do you think that that means that this implicates a persistent virus infection, or is it a downregulate? Is it causing a downregulation of a particular immune response that's detrimental?
Yeah, so Mark, that's both possibilities are currently holding up. It's, one possibility is just simply that the vaccine is inducing really good, immune response that clears any viral reservoir, in which case you can have like a permanent cure. Some people are getting a permanent cure from vaccine. Well, vast majority of people are not getting that. They're getting a temporal improvement in their symptoms, and it's not that all of their symptoms go away. So that to me tells me that potentially it's maybe cytokines or other factors that's induced by the vaccination that's sort of acting on autoreactive cells to shut down their cytokine production or potentially antibody production, but it's not a permanent solution. So we really need to understand like what's going on in these patients so we can provide a permanent solution.
Tina, could I comment on that in terms of related to antivirals? Really briefly, and I think the nice thing for one of these kind of forums is to take us a little bit beyond and not be afraid to sort of think and speculate a little bit, because I think we're also we're also deluged with data and information every day. Well, one thing for sure is that antivirals work best when there's a viral target. They don't work at all. If they're direct acting antivirals that are targeting the virus, they don't work at all if there's no virus present or if the disease is not viral related. And I think when, remdesivir was used late in infection, because at the time, of course, at the height of the epidemic pre-vaccines, the issue was, can we save people's lives? Can we get them off ventilators? And that fact that it was IV also encouraged that. And so I was actually surprised that it had any impact at all, because all the studies that have been done in Ralph's lab and my lab and others have shown that you use the drug early and it works, you use it late and it doesn't work. And that was true with both molnupiravir and remdesivir. And I've been thinking about this in terms of persistent virus infection, because the very first models at the lab I went in Stanley Perlman's lab in 1984, he was working on a model of viral persistence in mouse hepatitis virus, whereas if the dams were immunized and the infants were infected, they would not develop a severe acute disease, but they would develop persistent disease that led to chronic demyelinating encephalitis, right? So it led to demyelination of the brain. And that was the model that I was working on, mouse hepatitis virus, for all those years, which was really an immunological model of some ways, but it also was associated with long-term persistent infection. And so I think this is particularly interesting because you theoretically might have a place for an effective antiviral that decreased or allowed you to significantly decrease viral load over a long period of time that could be taken orally. So I hadn't thought of that actually until this moment. I'd been thinking prophylactically or very early in therapy, but there might be a role of that associated with, in fact, with immunization, because they're not mutually exclusive. So that's an interesting idea.
Yeah, I
And
love
then
that thought. I mean, what we need is a good clinical trial, you
Yes.
know? Yes, to
And I would
test that
even
idea.
say, Mark, adding to that, potentially another concept that could be explored in conjunction with antivirals could be FC region optimized monoclonal antibodies, optimized for killing of virally infected cells.
Okay, you have to unpack that. What does, what does that
Yeah.
mean? Those words, FC what?
So antibodies. So I'm an antibody person. Um, So monoclonal antibodies have two different major regions. They have the antigen binding region, so the Fab region, or the FC region, which is the part in the antibody that actually talks to the immune system. So a number of different recent papers from different groups have demonstrated that the FC region is actually required for not only protection, but you can actually um, improve the function of these antibodies if you if you modify them in a way that they can talk to the immune system a little bit better. So that could potentially be an additive therapy that you could explore, not just in the context of long COVID, for example, but if you were to maybe combine that with an antiviral. So now you have an antiviral that's targeting the virus, and you have a different type of biologic that's still antiviral, but it's actually engaging the immune system.
And so that would be different from the monoclonal antibodies that we've heard about, like the ones that are being used for early treatments.
Correct. So even without even without optimizing the part in the antibody that talks to the immune system, the antibodies will will do their thing anyways. So this would, like you correctly surmised, this would actually include, for example, the AstraZeneca cocktail or the Regeneron cocktail or the Bristol Myers Squibb cocktail. So this could potentially be, at least, in the small proportion of patients that Akiko mentions that could have symptoms, long COVID symptoms that are driven by the virus, then this could potentially be an additional therapy that could be explored.
Well, I do want to get all of your thoughts on what made this coronavirus into a pandemic virus. I mean, I've been writing a long time about infectious diseases, and there's been many different candidates, mostly different influenza viruses, avian influenzas particularly, and none of them have really become the pandemic that was promised. And then this one just sort of came out of the blue and suddenly it was everywhere. So, what have we learned about this virus? What makes it so special that it could be a pandemic virus?
I think the scary thing is that nothing makes it so special from other coronaviruses. Um,
No, I think, well, I'll wait till Mark finishes and jump in. Go ahead.
I'll let you go. Go ahead, Kanta, and I'll come back.
To me, the two big differences are from all the avian influenza viruses that have failed to start a pandemic, or SARS or MERS. There are two things. One is that this virus spreads efficiently from person to person by the airborne route, which did not happen with MERS or SARS. The second really big difference is that in both SARS and MERS, everybody who was infected had symptoms. And with this virus infection, we have a lot of asymptomatic individuals, people who don't even know they're infected, who are spreading the virus. So to me, those were the two game changers. I first heard about the person-to-person transmission at the very end of January of 2020. And I was at a flu meeting and I said, oh my goodness, this sounds like flu. This does not sound like SARS at all. And to me, that is really what made the difference in making this virus able to cause a pandemic.
I think Kanta and I were having these conversations in 2003. Is if SARS escapes, so we were watching it, if it escapes into Indian subcontinent or the African continent or in places where there were public health, we were concerned there might be public health, control issues in some places in the world, war zones, etc., that it would take off. And certainly you saw super spreading events, and we all thought that was going to be the one. But it had an Achilles heel, and that Achilles heel is it required symptomatic severe respiratory infection for transmission. And I think that one Achilles heel. The other thing is it's efficiency. So there was this issue of efficiency of transmission without symptoms, and second is that its R naught really never rose very high, except for these super spreading events. So those two things really made it controllable by public health measures that haven't been successful with COVID. And when I said nothing special about it, I shouldn't be cavalier about it. What I meant was that we don't understand in coronaviruses what would make one different from another. Right? There's no obvious signal except for the potential capacity of this particular spike protein to show this kind of dramatic variation. But when you look at it, it looks like another family member. You put them side by side, and they're going to look really, really similar. The number of nucleotide differences across the replicase gene is exceptionally small. They're highly related. And so what makes it unique, we don't know. And that's what I meant from the virological perspective. But I think from the perspective of the differences, we've been worrying, and all of our crying from the mountaintops was about MERS was, this is transmitted from camels. There's limited transmission from human to human, but if it gains the capacity for direct respiratory spread, it was maintaining a 30 to 40% mortality rate over about an 8-year period of people who got it. And we were concerned that this was one that would escape. And then MERS seems to have had the bad habits of both SARS-CoV and MERS-CoV, but we do not know why. And I think that's what is the important point.
And I think virologically that's true for influenza viruses as well. We do not yet understand what makes one influenza virus highly transmissible from person to person and another not. And that's been an area of interest and study because seasonal influenza viruses transmit very well. Avian influenza viruses generally do not, fortunately. And there are avian influenza viruses like H5N1 and H7N9 that have very, very high case fatality rates, but fortunately do not spread efficiently from person to person. So there's been a lot of interest in the flu community as well to try to understand what those determinants of transmission are so that we can then do a better job at risk assessment when viruses emerge in or cause zoonotic infections to know that this one is one to watch and contain immediately versus another that we could actually have small outbreaks and still not be at risk of a pandemic.
So one thing that I've written a lot about this year is the fact that this does seem to be transmitted through the air, um, whereas we didn't used to think that that was sort of the major route that respiratory viruses were transmitted, which to me, I guess now doesn't really make sense that they wouldn't be transmitted through the air. Um, but is there anything
Yeah,
that
yes.
else that we've learned that you think is surprising about that?
So this is an area that is fascinating, and it's an area where it's been very clear that virologists and physicians speak a very different language from aerosol biologists. They both know a lot, but they spoke a completely different language, used different terminology, different measures. And so the medical community basically said that anything that it was all based on particle size, and that if a particle was more than 5 microns in size that you emit from the respiratory tract, will settle out of the air within 3 meters, within 3 feet or 1 meter, and that's why hospital beds were always more than a meter apart. And that any virus particle that was under 5 microns could remain suspended in the air, could travel greater distances, and was then aerosol transmissible. Aerosol scientists tell us that is complete nonsense, that the particle size is more like 100 microns, not 5, and that particles actually, the size of the particles changes essentially as soon as you breathe them out. As they dry out, the particle size changes. So there's been and I think this last year there was a fantastic National Academy of Engineering workshop that brought aerosol scientists and engineers together with virologists and physicians and infection control people. So they're starting to break down those barriers and understand each other. So I think the science of how viruses or all pathogens are spread through the air is a absolutely fascinating field where lots has to happen. Because I'm hearing about built environments and how do you ventilate because normally we ventilate in indoor environments for comfort, not for health. Um, and so there's a lot that has to happen in that space. And I think the public health community was the slowest to the party. There was an unwillingness to use the word airborne transmission, even though the data was staring us in the faces. And so I think it had to do with a number of complex issues, such as if you label something airborne transmissible, then it changes the type of personal protective equipment that you need to then provide to people. And there was deemed to be a shortage of it. So how could you recommend that if you couldn't supply it? But I think it is time to sort of step back from all of that and look at the data, talk to our colleagues in other fields, and really break down some of these barriers and understand what is happening and how we can mitigate some of this.
Yeah, well, you mentioned talking to colleagues in other fields. It seems to me that science has gotten incredibly collaborative across fields and across the world. Huge numbers of scientists coming together to work on these problems. Um, what's your guys' perspective? Have you seen that, or are you seeing siloing? What's going on in terms of how this pandemic may be changing research?
Yeah, I think that's one of the biggest change that I've experienced personally is that, prior to the pandemic, we were pretty much doing things in silos as scientists. But with the pandemic, we've reached out to everyone who has the expertise and who's asking the questions that you're interested in, not just throughout the school or throughout the states, but throughout the world. So now we have multiple collaborations all across the globe, really looking at anything from immune pathogenesis to vaccine effectiveness to third dose, what is mix and match, many questions we can only address globally together. And that's been a very welcoming change for me. Like this team science is really the future. And I think the scientific community, like academic community, has to catch up with this in terms of evaluating scientists. The old traditional metrics about who's the last author, who's the first author, this kind of measurements is not conducive to kind of promoting this team science effort. And so that's something that I think the community, the scientific academic sort of environment has to change to accommodate.
Do you think it will last beyond the pandemic?
I hope so. I mean, once you do the team science, you can't go backwards.
I think if a lot of, I agree with Akiko, this has been fantastic, and some countries have done it kind of operationally at a national level, like the UK has done some phenomenal clinical trials and clinical evaluation by forming consortia. And so it's not just organically, I mean, it's happened because the government's thrown money at the problem. So I think the money that's been thrown at the problem has helped a lot. And I think my only worry about the longevity of this kind of cohesiveness is if money dries up and we end up being in a competitive environment again. So I think that, funding agencies, if they take away the funding as the reason to compete, then they'll be able to have much more collaboration.
I have seen the rise and fall from SARS and MERS. I think this is different because it's a pandemic. I think it's incorporating something. My question is, are the Akikos going to stay in the field? Are they going to stay interested in virology as the pressures on it become more competitive? And that wasn't targeted for you, of course, but it's because it's so important to have you in the field, and that we were a field that had one or two immunologists and two or three replication people, and three or four structural biologists, maybe, if we were lucky. And the question is, I think it's about infrastructure. It's like bridges. It's like, do you build them and then do you maintain them? Because I think the answer is correct. I've had more opportunities in the last year to talk to endonuclease and exonuclease biochemists, people who are developing small molecules, fragment-based screening, novel biochemical strategies that I ever have. I've had people reach out and want all of my MHV reporter viruses because they now want to look at the cell biology of coronaviruses in a BSL-2 model, and they want to try to understand issues of entry. And so that things that I thought I was going to be putting in the autoclave at the end of my career, I'm passing out around the world, and I'm thrilled. But I'll tell you, the most important thing, I think, is not for people like me to establish team science. It's to have the David Martinez and the other people who come into the field and who find the capacity to stay. And so what are the issues associated with that? Well, there's a couple major ones that I see. One is stability of funding, and that they can count on that. Two is that they find that that they can carry that work forward in a way that's meaningful if they want doing the hard work. So people ask me, how did you stick with coronavirus for 36 years? And my wife might have said, well, he's an idiot. That might have been the answer, but I think the idea is that the ideas compel you so much, and you're willing to fight for those ideas and go to the fundamental science. Because if everybody in the world is looking at the RBD on the spike protein, that isn't going to last, right? That's not going to last because people have to look at the more fundamental questions that kind of David is approaching. And the final one is, I think we just need young investigators, and we need to support them through the process and allow them the time to develop. and stay in these fields and make them understand that it's an area they can thrive in. That's what matters. Honestly, to me, that's the issue of continuity that matters the most.
Makes me almost feel a little bit guilty because I was a virologist and then I left to become a science journalist.
Yeah,
But
well, you may
here
have
we
left for very
are.
positive reasons, but a lot of people leave because they don't feel it's a sustainable career over the long haul, and that's a problem.
Yeah, Aaron became a science journalist doing your internship with us just at the point where the pandemic was hitting. So it was sort of the perfect person to have at the at a very bad time, but perfect for us. Which she was really instrumental in helping us properly communicate the virology about this.
Which we have done a terrible job. We've done a terrible job as scientists of communicating the terminology and doing it in a way that is recognizable, is understandable to people, and that engages them and lets them understand it's complex. And so it's not a contradiction to say one day we're developing a vaccine to try to prevent infection, and then we say, well, it didn't prevent infection, but boy, is it sure accomplishing the goal of preventing severe disease and hospitalization and death, and that science evolves and that it's not static.
If I could just add a point that perhaps hasn't been mentioned. So, to echo Mark, I would say, of course, understanding the fundamental biology of not just coronaviruses, but other viruses, I think is absolutely critical. But if I could just add, maybe an overarching theme that we need to rethink and maybe reinvent and redesign is how do we change our pandemic preparedness landscape? So instead of being a sitting duck, just waiting for a virus to emerge into a completely naive human population, I think we need to start prioritizing and investing more in pandemic preparedness on a more global level. Not just pandemic preparedness in terms of vaccine and monoclonal antibody or therapeutic design, but broad spectrum antivirals and even just public health infrastructure to be able to rapidly respond to a viral pandemic or a pathogen pandemic.
I mean, to get a little bit back to the idea that, science communication is hard. All of you have talked in some capacity to, journalists like us throughout the pandemic. What are some of the biggest challenges that you guys have faced in either communicating, your research or just talking about the science in general?
Well, so I use multiple different platforms to communicate. Twitter is the fastest. The moment I type, people can see what I'm talking about. I've also sort of engaged with more traditional media, newspapers, television, radio shows, and so on. And I actually decided in early pandemic to dedicate a big portion of my life to doing science communication because I think it's important for scientists to speak, from experts' opinion, what's going on, explaining to the lay public what is an immune system, what does antibody mean, how does memory work, all these basic fundamental insights that many people didn't have prior to pandemic. Now people are talking about neutralizing antibody titers and RVDs, and they have the lingo to be able to understand and talk about immune system as well as viruses. But I think that was important to me that we as scientists communicate with the public, and I think all of us try to do that. More than ever. Like I've never been in contact with so many reporters prior to pandemic. Occasionally, I would talk to them about this and that, but this has just completely changed the landscape. And I just hope that more people will engage in this because I think the society needs scientists' voice, needs our expertise, and needs people to explain to them, about what's the future of pandemic, how do we contain it, all these issues. So I think science communication is a big part of being a scientist.
And I do think that comes back to what David was talking about preparing for the next pandemic, right? Because at the beginning of all this, it was the communication was not amazing. And so, when the next pandemic happens, because it will, hopefully we'll be more prepared.
So I think communication is absolutely critical, and scientists are often not taught how to communicate. So I was at CDC and NIH and had a little communications workshops to attend, partly just so you don't embarrass the government. But University of Melbourne has one too. But I think, to me, probably what we could have, I think the public has been educated very much so in the last year and a half. mRNA rolls off people's tongues. They all know about boosting, and so it is amazing how much the general public knows of science now. But I think what we probably should have communicated better, and this is journalists as well as scientists, is that we were dealing with something completely new, and that the information was going to change, and that it is our job to be open to new information and to change our thinking as information evolves. That simply changing what we're saying doesn't mean that we are untrustworthy, that we have to find a way to communicate that trust should still be maintained and that we're responding to new information. So unfortunately, very often we are asked to give a definitive answer, and it's almost termed as if, if you've changed what you're saying, that the previous version was false, and it's rather that we're fine tuning it as we go.
I've give answers sometimes that are provocative and that the people don't like. They say, "How long is this going to last?" I'm going, "I don't think you want to ask me that." Or they'll say, "What are you working on now?" I say, "SARS-CoV-3," because we're trying to prepare for the next virus. And it's not really being glib. What the point I'm trying to make, and I tell people say, "How long this is going to last?" And I said, "Well, the virus will let us know," because. And it came back to some of what Kanta was saying earlier, or we were talking about earlier about the virus evolution. We don't know. We just don't know. And I think one thing that I knew, but I've learned anew, is that we need to have about 1 part of science and about 9 parts of humility around this. And that if we're in a position to tell people this, I start my things off. I'm going to give you a talk today. First, I'm going to tell you what I know, and what we've worked on, and what our results show. And I'm going to put those in context. And then I'm going to tell you what I think. And then I'm going to tell you what I have no idea, but it's my speculation about this. And I want you to put those in context, because the first is data, the second is my opinion, and the third is my worry about it. And so then I think that I've gotten responses back from that. I really appreciated that, because it allowed me to put things in context of what was really information and what was opinion, what was informed opinion, what was speculation based on what you know. And I think that is, it also says that in the face of a virus that is evolving independent of us, couldn't care less about us, that it's going to do what it does. And I've been shocked and amazed by its profound rapid evolutionary capacity. And that we don't know how long it took OC43 and HKU1, the human cold coronaviruses. We don't know how long it took for them to become the way they became. It's not going to happen overnight. And people want to say, well, in a year or so, this is just going to be a common cold virus. It's nonsense. It might be, but the statement is nonsense. And so I think that just this kind of humility we haven't had, and our willingness to say, I don't know, I don't know, I don't know. No one's going to keep interviewing us because they'll stop because we don't know. But it's the truth. It's the truth.
And I would say, adding to Mark's point about just saying what we don't know and having humility, I would think I would add that a strategy that has certainly worked for me in terms of communicating with the public is really trying to communicate the nuance in science, and really not speaking in absolute terms. So just coming with that humility of saying, just like in real life, nothing is, or most things most of the time, are never 100 or 0%. There's always a spectrum. There are nuances, and this is one of those things that is so new for everyone that we just don't know, and we're learning as we go.
Yeah, that is a good point. Thank you all for those thoughts. We are coming up on the end of our time, so I just had one final question because we wanted to end on a hopeful note after talking about such a grim, topic for an hour. Maybe very briefly, each of you could just talk really quick about what has given you hope over the past 2 years, like one thing that is your bright light at the end of the tunnel.
Well, what's given me hope is sort of the future. As Mark said, I think supporting and encouraging the new generation of scientists is what's giving me hope. And I see it in my own eyes, my trainees, as well as people around me who are young scientists who are now really engaging in the science of long COVID, science of COVID immunity. It really does give me hope, and they're so dedicated and talented and driven, and that to me is the future, and that's how we fight the next pandemic is by developing and growing and nurturing the next generation of scientists. So that's where I see the light.
The single most thing that has given me hope is just watch the community just come together, the scientific community come together with industry partners, um, to which we saw the development of not one, but three safe and effective vaccines in the US alone in less than 1 year. So um, to me, that's been one of the silver linings of this whole thing is seeing, if we let science take the front seat, then this is what we can achieve together.
Yeah, I would add that from my standpoint, it's the new technologies and the new team science and the sort of breaking down of barriers domestically and internationally. And so many people from many different fields have entered this field and have really contributed enormously. And that gives me great hope that we will see the end of this and that it will serve us well with future health threats.
Well, it gives me hope this particular virus is this stunning speed of development of vaccines and antivirals. People keep saying, why is it taking so long? Well, it didn't. I mean, even 7 years to develop, antivirals. I mean, and the fact that we now understand the targets, I think our capacity to target this is good, and it's going to serve us well, both from vaccines, antivirals, understanding of the immune response. All of this is coming together. It's overwhelming for those of us who try to keep up in any area, and that's why we need good science communication to help me find things to read that I haven't had time to read the primary literature on. But I think this, just the fact of the people you have on this, ranging across disciplines, across career stages, all focused on a common enemy, but a common area of science and understanding that could potentially lead us into the future and new models. I think that's the most hopeful thing.
I think we could all use some help keeping up with things that are happening in the COVID world. It's all moving so fast. But again, thank you all for taking the time to talk with us. This has been a fascinating conversation. I hope you all have learned as much as we have and how much our viewers will. Viewers, if you want to learn more about COVID-19 and the coronavirus, you can always visit our website at sciencenews.org and connect with us on social media. Thanks for joining us.
Happy anniversary.